passing out by that time. So, okay, are we ready? So, for this part, we're going to talk a little bit about some of the nuts and bolts of how you actually make a model, a rule-based model, and then after this section, and depending on how time goes, either before lunch or after lunch, Jose will be actually doing a hands-on demo with this, so that you guys can get your hands dirty making a rule-based model and seeing how that goes, and then he's going to demonstrate how to get that model from BioNetGen into MCell. So the basic idea is that you're going to be writing models in this BioNetGen language, which is pretty much similar to standard modeling languages for reaction networks, except that the molecules now have some structural components, which are just indicated by a comma-separated list. And the ordering of the components by convention, we usually go from N-terminal to C-terminal of a protein, but that's just a convention. The ordering doesn't really matter for any functional property. The components of the molecules essentially form a graph that comprise the network and comprise those contact maps that I showed. So you can think of these as meta nodes on a graph, if you like, where the components are the basic nodes, and then the molecules group those nodes. So for each binding site that you want, specific binding site that you want to have on your molecules, you can make a component site, and then some components may have also additionally post-translational modification site states indicated here, representing here unphosphorylated and phosphorylated forms. And one thing to note about the components is that they can be both binding sites and modification sites. And these states here, they're used to represent phosphorylation, but you could use different states to represent conformational states of a molecule or a component, a domain of a molecule, for example. And you're not limited to two. This can be you can have any number of different states. They are discrete in the current implementation. So here we've just made a translation of the components of our IgE receptor model into the BioNetGen language, representing the components and states that are shown there. And I think that's pretty much what I wanted to say at this point. Is that clear? You're going to put these definitions in a what's called a molecule types block of the model, where you're defining what the molecules are. It's very similar to the molecule block in MCell, where you define what your molecules are. Okay, that part should be straightforward. Right, set all of those things. Okay, so the next thing you need to understand in terms of how we work with the molecules in our system is how to create bonds between molecules. So an example of a bond is shown here. I have the IgE dimer molecule, which has two A states, and that's something I didn't mention on the previous in the previous description. If I give two components in the molecule the same name, it means they effectively act as symmetric components. And so here, in my IgE dimer, there are two binding sites that bind. There are two Fc portions of this antibody that bind to a receptor. So each one can form a bond to the receptor. And so I accomplish that functionality by having two sites that have the same name. So these are truly symmetric, and every aspect of their behavior in BioNetGen will take that symmetry into account. Okay, so we have another thing is that we have two different components named A on the receptor and components named A on the IgE dimer, and BioNetGen considers these as distinct. There's no problem of a namespace intersection here. The fact you can have different components with the same. You can have components on different molecules that share a name. Like SH2 is another common one that we would use, or Y, for example. It's basically its name is protected by the fact that it's a member of this other molecule. So that's allowed. What else do I say? So bonds. So bonds are denoted by the binding state of a component is referenced by this exclamation mark. And what follows that is an index that tells you what bond that link is referring, that exclamation mark is referring to. So the bond names are arbitrary. They're specific only to a particular complex, but they're just used to indicate the two endpoints of the bond. So here we have a bond between the ligand and the receptor molecule. It occurs through components, the A component of the ligand molecule and the A component of the receptor molecule. And the fact that they're bound together is indicated by having a bond with the index 1 starting here and ending here. Okay? So it's just, you can think of this as like a hyperlink, for example. You can only have two instances of the same bond in one complex. Two references to the same bond in one complex. Bonds can connect two components. You can't have a bond that connects three or more components. And every bond should also be terminated, so there should be exactly two references to every bond that's in your complex. Okay, so some other examples are shown here. Here's lin bound to the receptor, so lin molecule bound to the receptor, and it's bound to the, we can see here, the B component. So you notice in this particular instance, you have a component. Its state is referenced, so the component is in the unphosphorylated or U state, and then there's a bond that's referenced. It's bound through bond 1 to the U component of lin, for example, in this case. Does that make sense? So bonds are sort of the trickiest aspect of the syntax, getting that clear what that means. Another aspect of bonds is illustrated in this particular pattern here. In the model, we didn't actually include this, but in the extended, it's known that, for example, the lin kinase has a C-terminal region that can become phosphorylated, and when it's phosphorylated, it can bind to the SH2 domain of lin. And so you can have a bond that's inside, that is intramolecular within one molecule here. So that pretty much represents the complexity of bonds. Yeah, question?
Yeah. What does the A stand for?
Which one?
I mean, both proteins have A's. So
Yeah, so in this case, the Fc epsilon R1 receptor is composed of alpha, beta, and gamma homodimers. Okay. The A represents alpha, the alpha subunit. Here, the A is indicating that it's a cognate site for the alpha subunit of the receptor. It's just the nomenclature. Yeah. In the ideal, so in more advanced model descriptions, we would annotate these with protein references with IDs. What am I looking for? Like uniprot IDs, for example, to make references to specific molecules. So you could link this back to a database. So the names at that point become, they could be, the names are a little bit arbitrary, but you would like to know that this really represents what each of these represents in terms of the actual molecular entities here. This one would be a little bit tricky because IgE, this IgE dimer, this is an experimental construct where they chemically cross-linked two antibody molecules to make a particular ligand. It's kind of an artificial example. Not swimming in your body anywhere. Right. So yeah, the nomenclature is a little bit funny here. The U, but it is what is in where did I put this? Yeah. So lin, for example, it has a U domain. That's what it's called, the unique domain, and that's the standard nomenclature in the literature, which is separate from its SH2 domain. And the U domain in this case is known to associate with the unphosphorylated beta subunit of the receptor. When the beta subunit is phosphorylated, it binds to the SH2 domain of lin. So this is a weird, and which forms a strange kind of network motif here, where you have a weak interaction and a strong interaction. You have a weak constitutive interaction between Lin and the receptor. And then when the receptor activates, phosphorylates, the Lin phosphorylates the receptor, and it serves as an additional recruiting, a stronger recruiting mechanism. That's a positive example of a positive feedback. So, okay, I don't want to get too lost in those details. All right, so some rules. Now that we can make bonds, it's and we can refer to states, we can make the basic rules we need for this IgE signaling network. So here, this is the rule that we need to initiate the receptor binding to the ligand. And in this first rule, we have the ligand molecule is referred to as having two A sites. And when we refer to a component in a rule, or in a pattern where we're selecting a set of species with a particular property, when we refer to a component and we don't give a binding state, it means that we need that component to be unbound. So that's a convention that you just have to, that's one of the key conventions of the language. So here, when we say A, comma A, we mean that this IgE molecule has to have two free A sites. So it happens that it only has two A sites. So that means that both of the A sites of IgE have to be unbound. So this is the unbound, this is the example of this interaction, the unbound dimer binding to the receptor. So this has to have two free A sites, and this has a single free alpha subunit A site, the receptor does. And then they can form a bond, which is just indicated here. So I've underlined the components that are serving as what we call the reaction center, the part of the reaction, the part of the components, the part of the molecules that are being modified by the action of this rule. And everything that's left over, which is this other A component, that's context. So not all IgE molecules with a free A site can bind because we require both A sites to be free. So the second A site can be considered context, and then we're going to occupy one of those sites. That's what this rule is saying. And that corresponds to the situation where you have the ligand unbound to any receptor, which is actually freely diffusing outside the cell, now binding to a receptor that's on the cell surface. Okay. And that's that. And then the arrow indicates whether the reaction, this rule applies in both directions or only in a single direction. So here, and is typical with most binding reactions, it's reversible. So we're going to have this dissociation. As soon as you can make the bond, but the bond can also break. There's always going to be a rate for bond breaking as well as formation. And we would need, when we go to actually make this a reaction rule in BioNetGen, we now need two rate constants corresponding to the forward and reverse rate laws for this rule. Okay. How's that? Good? Are there any questions? Okay, so the second rule is an example of a phosphorylation reaction. So the action of the rule is to change the state of the beta subunit from unphosphorylated to phosphorylated. So this is it requires a kinase molecule, Lin, to be present in this complex. So this is an intra-complex phosphorylation reaction. It has this additional property that, and this has actually been verified experimentally by some clever experiments that were done by our collaborator to show that the phosphorylation within this complex involves not the receptor that the Lin is associated with, but the receptor that's adjacent to it in the complex. And so knowing that biochemistry, we can represent that in terms of a rule where the Lin that's associated with the receptor is required for a receptor that's unphosphorylated to become phosphorylated. So there's a lot of contextual information in addition in this reaction. Now, one thing you may have noticed is that this Lin is connected to this receptor, but we didn't say anything about the relationships between these receptors or the relationship between this receptor and this Lin molecule. And what this dot operator represents in terms of in terms of this pattern, this dot feature, is saying that all of these are members of the same complex. So whenever there's a dot between molecules, it means that there must be some path, some path of bonds that links this molecule to this molecule. So if we look at the actual way, the way that I drew this in a pictogram, we see that this receptor, which is this receptor, is bound to a ligand molecule, which is then bound to the other receptor. And that additional connectivity here is represented in shorthand by a dot. Okay. Now, but this dot is more general. So there could, in this particular model, there aren't, but you could imagine a situation where there are other mechanisms by which two receptors could be joined together into the same complex. And this rule would apply as long as you have two receptors that are joined in the same complex where one of them is bound to Lin. That may or may not be a desirable feature, but you should understand that that's a consequence of representing the rule in this way. So a more precise way to represent this rule that's specific to the way I've represented it here, this pictogram, would be to add the IgE. So we would add an instance of the IgE molecule. We could do it, for example, by putting a dot here, and then say that you'd have an IgE where one of the A subunits was bound to the A subunit of the receptor, which I would have to now reference, and the other one is bound to the other one. And so we could, and it's actually, let me briefly just write that out for the simple reason that it helps to clarify the bond notation just one step further. Maybe, yeah. So what we would have to represent just this part of the complex, we would have the FCERI, and we would have an A, and let's just call this bond 2. And we'll make this the Lin binding receptor. And then we would have the IgE A2, and a third bond that we have to reference now. And I'm just going to continue this on the next line. And FC, this is now our substrate receptor with a bond to this guy. Oops, I missed it. A bond here and an unphosphorylated beta subunit. And we'd also need the Lin. Okay, and now all of the molecules in my complex are connected to each other. And I've gotten rid of this implicit connectivity here. And it's good practice not to use this construct unless you absolutely have to, because it can create it can create mischief. You're basically making the computer do extra work that you might not want it to do. Being a little bit vague there. Exclamation refers to the binding state. The tilde refers to the internal state. So here the tilde represents, I'm referring to the internal state of B, and I'm saying that it's the U internal state, which represents the unphosphorylated state of B. Another thing to keep in mind here is that when I refer to a component, even if I refer to its internal state, if I don't say anything about its binding state, I'm requiring that it's unbound in that reference. So here, in this reaction rule, this phosphorylation occurs only if the substrate beta subunit is unbound. If something had come along and bound to this, like another Lin molecule, this doesn't take place, right? So there's an inhibition, there's a physical blocking of the kinase mechanism when that state is bound to something.
So as soon as there's a bond, this one is necessarily in a complex.
Mm-hmm.
But you can have a complex without an explicit bond.
Right.
It should be that there's some bonding that you don't make it explicit in the notation.
Right. It's tricky. What's tricky? The reason that it's bad practice to do this is it doesn't really affect network generation in BioNetGen very much. But when you do a network-free simulation, now, if you have two instances of a receptor in a complex, I have to trace all possible paths between them. So I have to do the graph search problem is bigger than if I restrict the search by making a specific path. Now, sometimes there may be a combinatorially complex number of possible paths, and I want the computer to do that work for me. But I should be aware that's going to, if I want that work to be done for me, it's also going to have an efficiency consequence to it. So it's going to slow down the simulation because the search is doing more work. So NFSim will slow down when I put patterns in like this. Sorry, not like this, like the one I replaced it with, like this, if I put these disconnected patterns in. Yeah, it has to check a lot more possible. It basically has to check all pairwise instances of receptors in a complex. No, nothing is ordered here. I could reorder everything, components and molecules. It's just the grouping that matters, and the bonds are named. I should say that the name doesn't mean anything outside. It's strictly for constructing it's strictly named for the purpose of uniquely identifying the graph components. But if I could this bond, I could call this 2 on this side. I could call this bond 2 on the product side, and it wouldn't affect the reaction at all. It's just describing the connectivity between them. Right. You could think of these as graphs. So Lin, for example, contains an SH, a U, and SH2. And the receptor has A, B, and G. And each of these has a state node associated with it. So these trees represent the sort of overall nodes on the graph, all the possible states of Lin and receptor. And I'll leave it there. And then you can represent a complex as this graph with a hyperedge between these two components. More questions?
So in the case where we don't specify the bond, it's still not order dependent. It doesn't make it. Like for instance, if we didn't specify the one in the top complex, does that know that the FCRI, the unphosphorylated B, is not bound to the Lin in any way? They're bound in that order, or it's a complex.
There's no order. So that's the interesting question. So this Lin, it can't happen in this system, but in principle, nothing here requires that this Lin is not bound to this receptor. It could be. It could well be. In fact, the one weakness of this formalism is it's hard to express not relationships. It's hard to exclude certain kinds of possibilities. We have certain ways to do it, but there's a balance between, this is a direct translation of mechanism. You have a picture that represents what can happen in a biochemical complex, and you can represent it as a rule. But to represent conditional thing, if you want to exclude possibilities, then there's not a single physical graph that it's derived from, and we just haven't made the syntax of the language easily handle that case. So I don't know. That's possibly a pathological rule. If you had the possibility to bridge between receptors, how would you exclude that? I'd have to think about it. It turns out that most systems you can express quite concisely in terms of rules, and we haven't adapted the language to include more expressive logic for the simple reason that we haven't had to. Nothing has forced us to do that. And that's a good principle to be conservative, because you can make a language too expressive, and you lose a lot of the power when you give too much flexibility. Yeah. I'll just leave it there. So good, yeah. So this actually, this reaction, these two reactions represent a lot of they bring up already a lot of the space of complexity that you can encounter in Rule. So there's not too much. It's a little bit tricky to understand the first, but once you understand this, you understand most of what's going on, I think. Yeah. Until we come up with another counterexample. Okay, so the parts of the rule, oops. That was worse for the people watching the video than it was for the people in the classroom. Parts of a rule. So we actually already went through this to a degree, but I'll just make it explicit. So we have standard chemical notation, reactants, some arrow products. The arrow indicates whether it's reversible or not. I should have mentioned on the previous example that our phosphorylation rule here was an example of an irreversible. This is a catalyzed transition. In order to get from a phosphorylated state back to unphosphorylated, we have a different enzyme, a different mechanism. It's a phosphatase. And it has different context. Actually, the phosphatase can act on this phosphorylated receptor regardless. It doesn't act within the complex, so it doesn't need this additional context, which is why we didn't make this reversible. So we have a separate dephosphorylation rule for that. Okay, so they're arrow, reactants, products. And then what we haven't talked about up to now is how we specify the rate law. So in this particular case, these k values represent constants that we define in the BioNetGen model. They represent the elementary mass action kinetic rate constants for the two different directions. So in the forward direction, we have a bimolecular reaction. This is a bimolecular rate constant with units of 1 over concentration, 1 over time. This is a unimolecular dissociation reaction. It's unimolecular. It just has units of 1 over time. 1 over, I think I said seconds before. But in one principle that Jose will introduce you to, in BioNetGen, the user is responsible for maintaining units, so which gives us a lot of flexibility. So when we, to work with different modeling languages that make different assumptions about units, but you're responsible. So if you if you inconsistently define your units, you're going to get incorrect results. So what we try to do for to make BioNetGen models reasonably comprehensible and to help people who are using this, we define sort of standard systems of units to use for different cases. And so for MCell, we're going to recommend a particular, if you're going to make a model that you want to be able to import freely into MCell, we will define a set of unit choices that you can make. So what units to use for bimolecular and unimolecular rate constants, for example, that will facilitate transfer of the models back and forth from MCell. MCell, as you should know by now, makes particular unit assumptions about what units different constants are in. Okay, and just to review now, so when we say reactants and products, so in the rule-based context, these things mean something a little bit different than what we're normally used to thinking about. And we've already talked about this, but I think it helps to go through it again. The idea is that these reactants are really patterns. They're selectors. They're going to select reactant molecules that have a specified set of properties. And there may be many sets of such reactant molecules in our system, reactant species in our system, that match the conditions here. And all of those will generate then the reactions that are specified. So for example, our reactant pattern, our IgE A, A requires an IgE molecule with 2 free A sites. That clearly matches just this free IgE molecule, and in fact, that's the only match it's going to generate in the possible space of in the space of possible species in our system. But the receptor pattern is a little bit less restrictive. It only says you have to have a free A site. So there are going to be many possible states of the receptor that have a free A site, and all of those can participate in the binding of the ligand molecule in this case. So here, the A site of the receptor is free, and the beta subunit is unphosphorylated, gamma is unphosphorylated, and neither of these is bound to another partner molecule. But here's another example of a receptor that is phosphorylated, and it also matches here. So the match set for the second reactant is larger than the one for the first, and it actually may encompass a large number of different possible species. And the basic philosophy of BioNetGen is that if you don't write it, we don't care about the match. So it doesn't affect the matching. So that means that I can, it's what we said before. The rules refer to the local requirements of the reactants, and it's possible to write very general, to have very general rules that have very few requirements. Most general rules will have the fewest requirements. So the process. So when a rule is active on a particular species, so here there are 3 different possible species, and there are 2 different sets. The free IgE can bind to an unphosphorylated receptor, or a phosphorylated receptor. And then what happens next? The transformation that's implied by this reactant to product transformation here is carried out on the reactants. So we take, in the upper example, a free IgE molecule, a free unphosphorylated receptor molecule, and we create a bond between an A component of the IgE and the A component of the receptor. And this is now referring to a bidirectional reaction. So the difference between a reaction and a reaction rule is that the reaction refers to specific species in my system, specific instances in my system. And so this says this particular, the free IgE molecule binds to the free receptor molecule unphosphorylated to form this complex, and with rate constants k plus 1 and k minus 1. And then, but this rule will also generate a second reaction, which says that the phosphorylated receptor can also bind with this exact same kinetics. Okay. So, and in general, the rule will generate many, many examples of these reactions in the network. We could figure out how many if we wanted to, because we can, by figuring out how many different possible states of B and G there are for a given receptor, and that number of reactions will be generated by this particular rule. But we don't need to figure it out because BioNetGen will figure that out. Okay. So this is sort of the core conceptual stuff. Once you get this, it's, you can go to town and make your own models, no problem. Okay, so the terminology, and I've introduced this before, but this is just formalizing that the part of the reaction that's transformed by a rule is called the reaction center. So most of the rules, the example rules that I'll write here, involve just a single transformation. You make a bond or you break a bond, you change a component state. But there can be rules that do multiple things, operate multiple changes simultaneously on one reactant set. So that part that changes is called the reaction center. That part which affects the change but it is not changed itself is called the context. Okay. Yeah.
So what would you do if you didn't care about either site? So it would be something like you don't care about either side of IgE, you just want it there and if it's present from intramolecular bond matches something very complex. So basically you don't care about either site, so don't change. You just want it present.
What's the rule going to do? That's what I'm
Like how would you set up the rule if you don't care about either site for IgE, for example? So both of those have to be free in this case?
Yeah.
If you wanted just one free, you
Mm-hmm.
just write IgA.
Yeah.
And then you don't care if either are free or not. You just want that species to be present.
Oh, okay, sure. Yeah, you could do that. Like that's its presence or absence is required, but it's not changed. It's like in that case.
Would it be like an empty set?
Yeah, you can. Yes, it would be. It would be this. A typical thing, something you might more often see. It would be weird to do this here and say, plus FC. whatever you're going to do. You're going to do something to the receptor. But this would be like it could be if you're going to do a catalytic process, but you're going to model it implicitly. You're not even going to bother to make the enzyme substrate complex. So this just transforms this. That happens, yeah. But so the way to refer to a molecule and not care about anything what's going on inside of it is this. Yeah, that's legit. What was I gonna say? Yeah, so more typically, it's weird for the IgE because it's not really catalytic; it's just a binder. But you might see you would maybe often see a kinase like Lin interacting with the receptor to carry out some phosphorylation reaction. You have to be careful. So when you make a choice like that, you're choosing to do a kind of that's an abstraction or a coarse graining. I mean, of course, the enzyme and substrate do form a complex. There is an interaction, and by making the kinetics here pure second order, you're eliminating possible competition effects. So I would generally say, especially in the context of spatial modeling, that would be a bad choice to make without some good justification. Like this is effectively constant. Maybe that's okay. I don't know. Yeah, but syntactically that's fine. Right, and this just shows. So I've said these things before, but it just shows it again. All right, we saw this before because I talked about that. There are lots of examples. So if you go, this isn't exactly a complete listing, but it's because it's not completely up to date. But there are a lot of model examples on our BioNetGen.org website. This model examples page has a number of ones that have been published in the literature. The BioNetGen distributions that come with RuleBender also include a lot of example models that you can look at, and some of those have been published. Yeah, question in the back?
So if we're not using this for MCell, what other simulators can you use it with?
You can export. Well, first of all, BioNetGen provides an ODE and SSA simulator. Both of those are pretty good. So for a lot of things you would want to do, those would be adequate. Our SSA simulator, it's not the fastest one out there, but it's good. But if you want to export to something else, and NFSim is very good. If you have a system where you need to be able to simulate a huge number of states, and you would have to generate an unwieldy reaction network, NFSim is a good tool to use. There's really only one other one that's comparable. But you can export any model in SBML format to pretty much any tool you want. Yeah, you can also, right, thank you. You can also export straight to MATLAB, so in a couple of ways. So you can there's a function called write_m file that generates just straight MATLAB, a straight .m file, straight MATLAB code. It's pretty convenient you can just run that, you can plug it in, you can do anything with that simulation module that you want. And the only disadvantage of that is that the ODE integrator that's native to MATLAB is not as good as the ODE integrator that's part of BioNetGen. That said, you can export also models to MEX files and do and compile them directly into C, and then link those to MATLAB, and then you have a very efficient way to simulate large biochemical network systems. And then we have actually some other tools. There's a tool called pTempest that a former student in the group wrote. If you Google that, I think there's a Google code site for it that lets you take. So if you export the models to MATLAB, either as MEX files or as M files, this allows you to do parallel tempering Bayesian Monte Carlo to do parameter estimation. So it's a typical problem. You have a model and you have some data and you know only a handful of the parameters for that model, and there might be 40 or 50 different kinetic parameters for your model. How do you estimate? How do you fit that model to data? So Bayesian Monte Carlo methods are really effective for that. And so we have code that lets you link to perform that on ODE models effectively. Doing that on something, stochastic models like the stochastic, that you simulate with either Gillespie or network-free or with MCell, the computational load for that is another order of magnitude or several orders of magnitude higher. So that's a custom job. But is that helpful? So you can, she left. but you can export files to SBML or to MATLAB very easily from BioNetGen. And it's not hard to write translators for other formats as well. But those two are then pretty much cover everything you'd want to do, I think. Yeah, to SSE, yeah. That's okay. So I was going to do, this I covered before we. Just check here. It's worth just throwing this up for a sec. So. So the idea, one key aspect of the architecture here is we specify the model in the BioNetGen language. And then once you have that, there are many different ways you can simulate that model. I've shown 3 here, and that doesn't include the export to MCell. There are different ways to simulate that same model that involve different assumptions. So ODEs involve continuum, the approximation that the system is deterministic, and you can represent all the variables as continuum variables. Concentration of all the species is a function of time. Gillespie, we include the effects of noise. NFSim is an alternative way of simulating a model that doesn't involve generating the network. And you can access each of these different methods by just a different command within the BioNetGen input file that you run on the model. So you define your model, and then you say what you want to do with the model, and you can simulate using any of these methods just directly within the framework. So that makes it very easy to say, okay, I'm going to do an ODE simulation of my model. Now, are there important stochastic effects? Is it really different if I do a stochastic simulation? I can take the same model and just do a stochastic simulation on the same thing and compare the results. Immediately I can get the answer to that question. There's some models that you can't simulate with either of these 2 methods, as I mentioned before, because the network is too big, and I can simulate that directly in NFSim. Okay, so this is just an example of how pathologically large these models can become. This is a model of the T-cell signaling network that's downstream of the T-cell receptor in the immune system. And the receptor is shown here. And these are all of these proteins that are involved in their phosphorylation sites are the proteins that are known to be phosphorylated within the first 60 seconds after stimulating a T-cell receptor. So, there was a mass spectrometry data set where they measured the phosphorylation dynamics of all of these different proteins. And they developed a model to try to see if they could match the phosphorylation kinetics with a biochemical model. And the state space of this model is, it's beyond computation. The only way you can simulate the model with this full wiring is using the network-free method. But they were able to do enough simulation that they could actually estimate many of the parameters for the model and get pretty good agreement between the experimental data and the model. And hopefully that will be published soon. That's not from my group. It's from my collaborator, Bill Hlavacek. Okay. So what I wanted to show today, what Jose will be working with, and I'll give a short demonstration now, is the development tool that we have for building and simulating rule-based models. It's called RuleBender, and it basically consists of an editor window that's got some rich text features in it. Shows you helps you with syntax highlighting to see the syntax of your model, and then you can visualize the model components through the contact map that's shown here, and then run simulations and view the output directly within the interface without having to go to external tools. It's also very easy to take a model that you develop in the RuleBender interface, simulate that from the command line with BioNetGen, or export it to MCell, MDL, SBML, MATLAB, and so forth, and then use other tools. So that's the goal, and the model in a nutshell is shown here. So what are the basic syntactic elements of a BioNetGen model? So I've talked about defining molecules or objects and rules as being the central focus of the BioNetGen model, but they're actually all of these different blocks, different components, and the ones that are in parentheses are optional components of the BioNetGen model. So there are parameters which define things like rate parameters and initial conditions, compartments which will define the topology of the different compartments in your model, and defining this is important for if you want a model that you want to export to a tool like MCell that's going to now give an explicit spatial representation. You need to define your molecule types. You need to define the species that are present initially in the system. Observables represent the outputs of your system. They're much like the counts that you define in MCell. Functions involve doing any kind of math on observables and also are used to define complex rate laws. And then the reaction rules, which define how these molecules interact with each other. And then finally, so this represents, these components here represent the description of a model. So this is a model of some biological system that's wired to give wired to be observed through these observables in this way. And then how I'm going to actually simulate that model is described in the actions block. One you can bypass simulation of the model altogether by just taking the model, generating a network, and exporting the model. That's one possible workflow. Or you can generate a network and simulate a model, or you can just simulate a model without generating a network at all. So as a basic motivating example, we'll just do the first couple of steps in this immunoreceptor signaling network that I've been defining, which is just the binding of the ligand molecule to a receptor and the formation of a dimer when the ligand that's bound to a single receptor then binds to a second receptor. This process is called cross-linking. So, and this in a typical experimental situation, you have a cell that's maybe in suspension, and it's interacting with freely diffusing ligand molecules that are in the cell in the suspension, and then the binding occurs to a single receptor, and then once that first binding event has occurred, the ligand molecule is now tethered to the surface, and the second binding is effectively a reaction between two species that are present on the plasma membrane. So if we think about this in an MCell context, the first reaction involves a volume surface reaction, and the second reaction involves a surface-surface reaction. And we can see that between the first step and the second step, although both involve formation of a bond between the ligand molecule and the receptor, the effective dimensionality of the reaction volume has been changed, has been reduced from 3 to 2. And so this second reaction occurs in a much smaller volume than the first, because in the first, the ligand is diffusing all over this three-dimensional volume, and in the second, it's confined to only be moving on the surface of this cell. So that's going to result in a rate enhancement, an effective rate enhancement, which we can see. Hopefully, Jose will explain that to you this afternoon. So these things are always a little bit confusing, so that's partly why we like to do them. If you can handle the confusing cases, then the non-confusing ones are that much easier. Okay, so what we have to do to do a compartmental model specification, and I'm just going to illustrate here the details. Jose is going to drag you through the details, so we're not going to go into a huge amount, but I just want to show you what it looks like. So we need to define this this topology rather, that's going to correspond to a specific geometry, but we're making very sort of abstract assumptions here, which is just we have here a system of compartments. We have you'll notice that every membrane contains a volume, and every volume can contain multiple membranes within it. So that basic structure forms a tree structure we can use to define an arbitrary geometry that we would encounter. So here, the whole reactor is contained within a 2D compartment that we call the wall. So you just think of the walls of our beaker. This is actually not really, this is necessary now for the translator to specify, but in general, we don't normally think about this for most BioNetGen models. So the outermost compartment from the point of view of chemistry taking place is where the ligand molecules are located in the extracellular region. So outside the cell is a 3D volume that molecules diffuse in. So the second this number here, so we have the name of our compartment, the dimensionality of the compartment, the volume of the compartment, and then what its parent is in this tree. So the extracellular compartment is contained by the wall, okay? Which again is, and the wall doesn't have anything outside of it, so this entry is blank. But every other element in my compartment topology has to be contained by something. So the extracellular space is the area outside the cell. The plasma membrane divides the extracellular space, lies between the extracellular space and the cytoplasm. So it's a two-dimensional compartment. It also has a volume, and I'll say something about that in just a sec. The extracellular is what contains it, and so forth. And then the lowest compartment in our tree is the cytoplasm. It's a volume. It has a volume, and it's contained by the plasma membrane. So this is how we can generally specify compartment topologies in BioNetGen. And there's more, you're going to learn a little bit more about that, and there's also more detailed documentation about that on the BioNetGen website. But the key thing that I want to say here is that when I mentioned this reduction of dimensionality for the surface reaction, so the way that we handle that case in BioNetGen, the way that we handle surface reactions, is to consider that there's essentially a surface volume that reactions within that occur on the cell surface are occurring just in a reduced volume that's basically the area of that surface times some thickness, which is effectively the corresponding to the dimensionality of the molecules that are fluctuating on these membrane surfaces. So a typical dimensionality for proteins that are tethered to a surface is something like 100 angstroms. And so the thickness that we typically estimate for molecules interacting at this surface layer is about 10 nanometers. So you can think of this in different ways. In some sense, it's a fudge factor. Surface reactions are constrained by other things than pure volume considerations. But this gives us a pretty good handle on unit conversions between, and how to estimate 2D reaction rate constants from three-dimensional ones, which is typically what is measured in an experiment. So the convention here, so when we specify volumes of a 2D compartment in the BioNetGen compartment list, this is really the area times some effective thickness. We can vary the thickness. The thickness could be different for different membrane layers in our system, and we can do other fancier things to differentiate these kinds of reduction effects for different reacting partners. But this is just sort of a simple way to handle these effects in the absence of other detailed knowledge about what's going on. And the problem is, for those of you who've tried to construct some of these models, there's just often not a lot of data constraining or telling you, how to relate 2D to 3D reactions. So what we're trying to do here is just to define a simple system that will work to give you sort of, I don't want to say order of magnitude, but good, reasonable approximations for the behavior, reasonable starting points, which you could refine using parameter estimation, for example. I mentioned that we have a system for now taking a model that's defined in this way and exporting it to MCell, and that's what we'll be doing this afternoon. And the key is just to be able to define the model in the way that I'm describing here. And what are shown here, so this is we've applied this pipeline to the IgE receptor, this immunoreceptor signaling pathway that I mentioned before, that has a very high, 354 species and thousands of reactions, and we can do those simulations effectively in MCell. This is just a snapshot of a simulation using this translation, and it typically takes maybe 30 seconds or so to take the model from BioNetGen to export it to MCell and then start a simulation. We can get, here, just showing here, that under the right conditions, we can get good agreement between the spatial simulation and the well-mixed simulation, but that's going to be dependent on rate constants. As the diffusion slows down, you may see departure from that agreement. I'm not sure that's what's showing here, but I'm not sure what the numbers correspond to. You can ask Deepak for more details if you're interested. All right, so let me just stop there and ask, are there any questions up to this point? I was going to give you a quick showing of RuleBender, just to show you how that works. But I'm almost wondering if we should. Yeah, I'll take you on a tour of the interface before lunch, and then we can do that. We can start things again after lunch. But I'll stop for questions now. If you.
Maybe I missed it, but are you only using the information that's out there, or are you also like using machine learning to fill up the gap?
Machine learning.
I mean, what if something is missing in between that we don't know about and it affects the model?
It could, yeah, sure. We don't have a way to, I mean, if a biochemical mechanism is deficient, how would you determine that that's the case? Yeah, well, the simplest way to determine that is you make a mechanism, you have some data that's probing that biochemistry, and it doesn't fit the data. So that's a good starting point. Now, how when it doesn't fit, is there a systematic way to infer what's missing? There is some definitely research along those lines. So one idea is to say, if you have a model of some, sorry, I went off camera, but I need to sit down for a minute. If you have a model of some, for example, our IgE receptor network, and you can easily find now, if you go to a database, many different protein-protein interactions involving those molecules that are not modeled in your system. Is there a systematic way to pick out the most likely candidates to increase the complexity of your model and then try to include those in a fit? I can't. I know that people have tried things like that. I mean, the short answer is there's no part of BioNetGen that addresses that question directly. But there is research in that area. But the connection between the mechanistic modeling tools that we have now and databases of protein interactions or databases of molecular biochemistry, that's to be done. That's the area of research interest.
I've seen people try, for example, establishing reaction networks as Bayesian networks, and see given the known biochemical information, can I get a group of conditional probabilities and establish that as a correct Bayesian network? I mean, that's not the case. Well, maybe that's indicating like a string, right? So that's what I've seen people do. I mean, it's limited in what you can try, but that's definitely the direction.
Yeah, but I mean, reaction networks are not Bayesian networks. That's the biggest problem. They're not even closely approximating. So that's a big problem. Yeah. But
basically, this is the optimal way to construct a model from all the experimental information that's out there. Can I take that as
that's a good. So that's interesting. Do I want to claim that it's the optimal way? it's I want to put another constraint on that because I mean, Jose is talking about it. There may be ways to, if you think about a Bayesian model, it's a completely different idea of how, there you basically have a graph on the molecular, the typically framed as a graph on the molecules that are part of my pathway. And I just, my initial reaction to that is blah, because like, look, what would that look like? What would the Bayesian network model look like? You know what I'm going to show, right? This. What does it mean to say that, to say, okay, here's my first node, and here's another node, and here's another node, and I'm going to take all of this, and I'm going to make one big arrow between them, and that's going to be and another arrow here, and maybe there's, what is the Bayesian network description of this? This is real. We know this is happening. So how do you describe that as a Bayesian network? So we're going to abstract that to. I'm not picking on Jose. I don't know. Pick another molecule here. All right. So wonderful. Anybody who knows these molecules, this is not a very informative model of a network, of this particular network. I mean, in some sense, information does flow this way, but is that really how the system is regulated? No.
Try to model it, how the system is regulated. Try to model information necessarily.
Yeah, but we're interested. So we're interested here in biochemistry, and what's interesting is that much of the biochemistry is known. If there are people who spend inordinate, their lives mapping out the details of what post-translational modifications occur, under what context those translations are important, what molecules are responsible for mediating those transformations, and so on and so on and so forth. And the question is, does that matter? Does that matter for the outcome of signaling? In some cases, it will, and more. But if my model is missing some of that knowledge set, how do I systematically add those back in? And I would say that's, like I said, we've just I would say in the last couple of years developed tools where we can effectively put this all together. So that now if I say, okay, I have a model that includes these guys, but not some additional layer. Which ones do I add? That's the next step. Yeah. All right. I'm getting. I don't want to get too bogged down in the question, but it's at the heart of the matter. Yeah.
But an alternative way of doing it is exactly the parameter estimation.
Yeah, that's another. But that's not going to. So the idea is that if you do parameter estimation, you say, okay, I have a model, and I want to find out is this model consistent with data, and what parameterizations of the model are consistent with data. And you could get a null answer. You could get an answer, no, that this model is not consistent with the data, period.
Or the parameter estimations are not realistic.
Yeah.
They're either too constrained or they're way out of line.
Yeah. So in that case, something is wrong with the model. But what's very hard to do is say, and people claim, there are people who have done studies where they claim to be able to do this next step. That's why I'm not saying it hasn't been done or it can't be done. Where now that I have a deficient model, do I have a system that predicts what things I should try to repair my model? There's some work out there on that recent. But the question is whether it's that you can get to something that's at the resolution of this at the same time, and I don't think not so far. Tom.
At the fine scale of the biochemical kinetics, there's a thermodynamic principle called detailed analysis. And microscopic versatility, where the rate constant going forward and backward around a closed loop have to satisfy certain mathematical constraints in order for delta G around the loop to be zero.
Right. That's an area where of model building where you have to be careful. Once you make sufficiently complicated models, you have to be aware of detail balance and to enforce those, to make models that are consistent with that. I'm going to get up in a second. Right.
But
Right.
it could be part of the constraints you're trying to
It could.
specify in determining whether your model is completely consistent and sufficiently
Sure.
consistent.
But
But certainly isn't the only.
it's not going to be the solution. If you have a model that you haven't taken care of to satisfy detail balance, but it's not consistent with the data, then making it consistent with detail balance is not going to make the model consistent with data because it's a subset of what you already searched. So you need something is missing. So I think the fundamental, and if you ask a biologist, what's the most, what do I want models to tell me? They most want to know what's missing from my system so I can go and find it. Because that's, after all, the fundamental problem of modern molecular biology is to find the missing pieces, right? That's all, it's what it's all about. It's the search for missing things. So if they're not that interested, many of you may be molecular biologists, but in general, collectively, culturally, people are not as interested in the problem of, now that I know what's there, how do I understand how it's all working together to give some outcome? Most of what's going on is, how do I find the next missing piece so that I can find a molecule that is mine, that I build my career on? That's how molecular biology works. So they're not so interested in, if I are, all of these components are well known, right? They're not so interested now in trying to figure out how this is all operating because they're all known. So I want to find the things that aren't known. Okay, that's a problem. That's a problem for all modelers have to kind of confront, in especially in your interactions. I see so many nodding heads. It's so frustrating to be told, well, what can you tell me that's missing? I'm like, I can't tell you anything about what's missing. I don't know. Those are the unknown unknowns, right? Those are the things out there that are just lurking and completely fry any hypothesis I have about how the system is behaving. So, anyway, all right.
John Guggenheimer, a couple years ago, published a paper that says it's a fool's errand to try to simulate a subset of a reaction embedded in a whole.
Right.
To a certain degree, that's true. You can only really do separations of things at clean separations of timescales. Make one part of the model and another. But if you've got two different parts of a model that are operating at the same timescale, and there's some connection between them, you can't separate one from the other.
Right. So that's a big problem.
Although I would think one thing you could attempt to have a model for this thing was not to identify molecular pairs specifically, but concept.
Yeah.
Say, based on our analysis, there's something missing here that
Yeah, I mean, things get interesting as soon as the model doesn't fit the data. Yeah.
But it's like there's something missing, or some black box here that you need to make this work.
The best part, though, is, what's missing. So if I say of this picture, what's missing here? What's not here? Yeah. What's amazing is it often doesn't work. Even at this level, it often doesn't work. Then things get interesting, and I think that's what we're after here. But okay, I think if you take us in a more philosophical direction, I'm just going to defer it. But if you ask a specific question.
I just have one question.
Mm-hmm.
Is all the pieces that you represent here is for this particular problem? Is it all? I mean, are each of these elements that you depict in the system, are they experimentally? I mean, have they found each individual label that you mentioned over here?
Every single one of the components that's here is either a post-translational modification site that's been observed in the experiments that were used to calibrate this model, or known interaction domains of those proteins that mediate interaction. So this, although this looks horrible, this is very, if you go to, every one of these molecules is very well systematically studied. They've made knockouts of these knockout mice with these proteins knocked out. They've then re-expressed many different constructs of these molecules with different sites, mutated or not mutated, and looked at their behavior and signaling outcome. So there's a huge knowledge base that this is all built on top of, and which is why what the idea of the rule-based approach is to say, take what we know about biochemistry, express that in a model, and be able to simulate that model. That's the fundamental question that we're trying to address. All questions are built on top of that. If I go starting from what's known or hypothesized about biochemical mechanism, what can I learn? Okay. So this is all real. I mean, there are no made-up things here. There are no theoreticians' constructs here. These are all.
So how does this model then help you? So I mean, so if you have everything that you know.
I'm not going to answer that question.
So going back to your missing protein, could you, if you have something that's happening and you know it must be mediated by a protein and you don't know which one, could you put in a black box protein?
Yeah. So that's a common trick. Yeah, that you say we think the model doesn't fit as it's specified. If we add this interaction, now it fits. That's a sufficiency test. That's good. Now you have now you've said to your experimental collaborator, colleague, go and find me this molecule, and you're in good shape. So that's what you can do, and it's possible. It's quite possible that we already know. So if you got that answer, then the next step would be to go to a database and say, what are the things that are known to interact with this protein that could then be this mystery molecule. And that's a feasible bioinformatics kind of question. I'm not sure if it's a machine learning question, but it's a bioinformatics-y kind of question. Machine learning would be like, predict unknown interactors of this protein based on sequence, or whatever you want, whatever characteristics you want. But yeah. That's good. Okay, I'm just resisting the urge to say anything else. Let's just do a little bit of a walkthrough of RuleBender, and then we can have lunch. I can feel that coffee that I had and my blood sugar going down at the same time, just making me. So hopefully, that's why I don't want to make any grandiose statements in that state, because I'll regret it probably. So let me just quit this for a second, and I'll restart. So RuleBender is an application. So we have a website, RuleBender.org. It works good. Where you can get the RuleBender tool. You click on downloads here, and you get some information about some publications that have been made on RuleBender, and you can download the latest version, which is here. And I just wanted to show you, so it's as simple as going to the Google Code repository and clicking on the right version here. And what I just wanted to mention is that this RuleBender distribution that you get includes the interface RuleBender, includes the latest release version of BioNetGen and NFSim. So all the tools that I've been mentioning today, you can get through RuleBender.org, is what I wanted to say. That said, if you want to get more information about BioNetGen, the best place for detailed information about how to make models in BioNetGen is at this BioNetGen.org website. All of these links, by the way, are available from our MMBios, so you can get to all of this stuff. It's a little bit of a mess. We haven't put everything under one umbrella, and that's why I'm just mentioning some of these things now. But there's a lot of information, a lot of documentation here. Special tips are also worth checking under here that give you help with specific modeling questions.
Show the first one.
Yeah, you just put that there, right?
Yeah,
Okay. All right. It says it's new. I don't know that one. Yeah. So there's an information overload here, but it's if you're interested in this in rule-based modeling and you want to get more into it, this is a great source of information. It's better organized than it looks, but and the other source is that we have a mailing list that you're welcome to join and ask questions on, which is the link is up here. So okay, so once you've gotten RuleBender and gotten it installed, it's pretty much out of the box installation. It comes, all the links should be set up, or there are there's installation directions. But I think on all the major platforms, it all comes configured out of the box. Then you're working in, this is the main interface, the main window, and this is built on Eclipse's rich client platform, so it's very much an interface that's very similar to Eclipse, if any of you guys have used that for code development. And the advantage of that is that it makes it very easy for us to make new capabilities and build in new functionality into this interface, and we get a lot of things for free, like context highlighting and so forth. The disadvantage is that the learning curve for the workflow is, there's a little bit of a learning curve. It's not Blender steep, but it's not completely intuitive out of the box, because it's a little bit old-fashioned in some ways. So I thought it would help to just walk through what's here. So the navigator that's shown on the left-hand side here are all of your project folders. So you can see I have for different projects for different things. Those are where I'm going to put different models, and I group them together on some theme, like my very special lab project. This was some one I used for a demonstration for a lab where we started to make models. So the first step that I wanted to show people was I make a project folder to keep everything in, to keep things organized. So underneath each of these, and I have one for, we'll just go to this one. We have different models. So the BNGL files are the model files that we are using. And they're the BioNetGen language files. So those are the primary files that I want to open to work with in the interface. And you can see, so I double-clicked on one of those files, and we come up with a model description file. This is a BioNetGen language format. And oh, there's a little bit of a glitch here. Let me reopen that because I want to show you something. Okay, so there's a slight trick here. So on this side are different tools for visualizing and simulating models, and it's helpful to have this contact map tool open when you open a model, because then the contact map will automatically display. And there's a quirk that if it happens not to be the one that's in the foreground when you open the model, the contact map doesn't display, and it doesn't come back. You have to reopen the model, so it's just a weirdness. But most of the time it'll come up this way, and so we can see that we have a model that involves two different molecules, and a ligand molecule and a receptor, and the ligand binds to the receptor. We can even browse into this, into this model a little bit more in depth, but we'll leave that for now. And what's interesting, so I can change the layout of this map. I like to make the molecules go horizontally across, and the connections connect hierarchically down, which makes for a scale, a representation that scales well as I add complexity to the system. And that layout will be preserved. So it's the coordinates of the different molecules are saved in this pos file, and when I reopen this model, the contact map will come up just as I laid it out. Except that it might, the position of the whole thing might get shifted around, but the whatever layout I do here will get preserved. Okay, so now we have our BioNetGen model, which has the different blocks that I described earlier, parameters, compartments, molecule types, species, observables, and reaction rules. And then And those are all enclosed in a begin and end model statement. So this comprises the BioNetGen model, the part up above line above line 49 here. And then everything after this is what I'm going to do to simulate the model. So here we're generating a network, and we're going to run an ODE-based simulation of this. Actually, no, we can change that. An ODE-based simulation of the model. Okay. And it's pretty much to generate. Once we have defined the model, to run simulations is pretty much just this simple. I can click over here to this play button, and this is a very primitive interface for just running simulations of the model. So this is going to just run BioNetGen on the model that's specified. I have to click over here to populate the file button. It's just and then I hit run, and it's going to run a simulation. The output of that, the console output of the simulation is shown here. I better do this. Trying to, right, so I can see what happened when I ran BioNetGen on this model file. So it reads in the model, it generates a network, and here are shown the different iterations of the network generation, generating at each iteration species and reactions until I don't generate any more reactions, and then we stop. It gives some summary output. Tells me I wrote the network, and now it's going to do a simulation of the network. And looks like there's, okay. And then this is the actual simulator running, reading the model back in and running the simulation, and it finished. And the whole thing took the blink of an eye, basically, because this is a very small model. And then we see that what came up automatically was a plot showing the concentrations of the different observables of the model that I defined as a function of time. So the observables are what we defined as the outputs. They correspond here. The observables that we made correspond to the free receptor molecules, which are shown in red. So you can see that initially there are 400 free receptor molecules, and those are decreasing with time as the ligand binds to them. So the ligand bound, the receptors that are bound to ligand are shown here. They're increasing in time, and then receptors that are bound to ligand can then bind to another free receptor to form a dimer, and the dimerization as a function of time is shown here in green. Okay, so and we can, this plotting interface allows us to look at whichever of the observables we want. We can zoom in, look at specific portions of the curve. You can change the axis scaling to get log plots, and it's not so useful for time dependence, but it is useful for when we can do a dose response curve. So that's a basic walkthrough of just running a simulation. We can do other things. Let me see here. The one other thing that we're likely to want to do to this model, and I have to think about it, we can easily do a parameter scan. So we want to know how does a given parameter affect the model behavior. And I just have to look at this for 1 sec to make sure I know what I'm doing. So that we could scan, for example, the number of ligand molecules that are present in the system. Here we had 6,000, and we see that that the steady state, approximate steady state value of the number of dimers here is significantly less. So the maximum number of receptors that are in dimers is something on the order of 75 here. So presumably if we increase the ligand concentration, we're going to get more. If we decrease it, we're going to get less. So let's just see what happens if we vary the amount of ligand as input. So the parameter we want to change is lig_tot, and we can vary that from a minimum of, say, 1 to a maximum of, say, 600,000. And we can look at 20 points. I hope this works. We'll see. Sometimes and a key thing, so often when you're trying to understand how a system is going to behave, you can see I picked 5-decade range of concentration here. So if I vary that in linear increments, it's not going to be all that informative. So I'd better vary that over a log scale range. So I click on that, and then I need to pick a simulation end time. A good practice is if I want to look at a dose response curve. So how does the output of the system depend on the initial concentration? I normally want. I mean, I could be if I'm thinking about certain experiments, I might be looking at a fixed time after simulation. But very often we're mostly concerned with what's the steady state value. So I would want to make sure that whatever the time I'm going to pick for my simulation is long enough in the future that I've reached steady state. And I should normally check that for at least the extreme values of the range to make sure I'm at steady state throughout. But I'm lazy. I don't want to do that. So I'm just going to pick what I think is a long time, and we'll see what happens. This number of time points doesn't matter because we're only going to look at the endpoint for our dose response curve, and we're going to use ODE. And then we just hit run here. So I'm trying to give you the general principles here rather than get a chance to do some of the hands-on stuff later. So the details aren't as important as just the overall picture that I can use a parameter scan to look at how changing a variable of my model is going to change the behavior. And you can see stuff is happening, but it's awfully hard to see. And the only thing I can see really well, I can get a sense of here is this green curve. Surprisingly, this is number of dimers, receptors and dimers, and it's going down at least over this range that I can see well on the linear plot. So when we're varying a parameter over many decades, we really want to look at it with log scaling. So when we do that, we get something interesting here, which is that there's a regime where increasing the ligand concentration increases the number of dimers. I don't know how well you can see the green curve, but maybe if I get rid of these guys for a sec. So here's what's nice about the Eclipse interface, this RCP, is that everything is configurable. All these windows are changeable. Just like in Blender, you've got a lot of flexibility. Sometimes windows will jump in ways you don't expect. So part of learning the interface is learning to deal with that. But what we can see now very clearly is that at low concentrations, we increase the ligand concentration up to a certain point. We increase the number of dimers in aggregates. But then at higher concentrations, as we continue to increase the ligand concentration, we get fewer dimers. So if dimers were required for signal propagation, this system would have an optimal range of ligand concentrations to maximize the response, and clearly at very high concentrations, the response is going to get attenuated. Has anyone seen this? Know why this is happening in this particular system? Offhand, it's a little surprising, right? You'd think if I increase the ligand, I should get more receptors bound, and that should increase dimerization, which works to a point. So my ligand is bivalent, and it's binding to receptors to form these aggregates. So these are the receptors, and these are the ligands. And what's happening here at high concentrations, you form lots of these complexes. Okay, so you form lots of bound receptors, but this complex, in order to make a dimer, it needs to find a free receptor. Okay, so this mechanism is competing with itself. The more of these guys I make, the fewer of these guys there are, and the fewer dimers I can make. So this is a phenomenon that's well known in multivalent processes. It's well known in immunology as the so-called precipitin effect, because antibodies are bivalent in the same way as this ligand is in this system. That you get this what's called high dose inhibition. Very high ligand doses turn the signal mechanism off. So, and this phenomenon has been comes up in many different signaling contexts because you have many proteins that can bind to multiple proteins simultaneously, and they can give rise to these kinds of so-called precipitin effects. You're dying to say something.
Well, seems like if the reaction is reversible,
Mm-hmm.
then the dimers are more stable than the half-filled thing. That eventually, if the half-filled thing reaction is reversible, then when the ligand comes off of one, it could immediately find one that's that it can make a dimer with. I mean, the dimers are more stable than these things, and over time, eventually. So I think what you're saying here is you're not at steady state yet.
Oh, let's test that.
Okay.
How long do you think we need to get to steady state?
Well, it might, the affinity of some of these things is like,
So the off rate is 0.01.
so it could be a week of simulated time.
A week?
A week of walk time.
So what's the? Hold on. Let's test that. So the off rate here for the dissociation is 0.01. So that means that the typical time scale in the system is like 100 seconds. But okay, I'll grant you that could be the time to reach equilibrium could be several orders of magnitude longer than that. So let's just conservatively say that if I simulate for 1,000,000 seconds, is that good enough to see steady state? We can test it if you want to do, but I can. So okay, so the beauty is, and so here I'm making a little bit of a case for why wouldn't you just make this model an MCell to start with? And the answer is because you should first have a basic understanding of your system and what properties it's capable of exhibiting. And you can test that over a much wider range of parameters when you're using ODEs than you can when you have to do a particle-based simulation. Same reason I wouldn't, don't start with the most complicated model of the system you can have. You really need to understand some of the fundamental concepts acting. So the key idea is I can run a simulation that's what did we say before? That's 1,000 times longer than the one I ran before to test our steady-state hypothesis, which we couldn't do it in a spatial modeling context without burning a lot of cycles. Or using, so it's good to work through some of these things sometimes in advance of doing a brute force simulation. So okay, here I'll press run. Let's see what happens. Okay, so is Tom's conjecture correct that this is an equilibrium problem and we just didn't get to steady state? And I'm glad you said that because it's not obvious. But the answer is no, it's not correct. We were already pretty darn close to steady state. Let me just take this out. So here the curve it's converged. At 1,000 seconds we were pretty well converged. I mean if we I can actually just show you. Let's see. A little bit of way of showing off the interface. Ooh. It's showing off and at the same time showing the limitation. No. You should be able to split the window. Do you know how to split it? There's a way. It's going to do. All right. See, that's not very. You can see sort of that they're pretty much the same. In this view, but it's suboptimal for comparison. I want to do a side-by-side split. It's somewhat. Some of the problem I'm having is because of the screen resolution. Yeah. So ideally, yeah. All right. What did I do here? Ooh. So I'm showing you the pitfalls of Eclipse interface here, the rich client platform. I can get back. No. See, it's getting close to lunch, and I'm forgetting all of the tricks as I haven't used this as much in recent days as I usually do. Oh, I don't know. I give up. I've gotten myself into a state that I don't know how to recover from gracefully. And all of this is because on this with this screen resolution, things don't behave quite as nicely as they did before. So what did I do? I have to. I'm going to quit. It's like Blender in that you fall once you think you're riding the bike and then you start thinking about what you're doing, you fall off the bike and you have to remember how to get. Now, this didn't fix my problem either because it very nicely saved the horrible state of the system that I managed to get to. There's a way. Do you know how to get back to the? I want it to go to default, but it's not. All right. Maybe that's a good place to break for lunch. I tied myself up in a knot. So what I was trying to do was to show you the plot side by side, which is relatively easy to do. You just have to drag it in the right place on the screen, but somehow that wasn't lighting up in the right way. And as soon as then I tried to do other things, and I've now gotten myself into a bind. With the one click, I can get out of it, but I have to remember where to click. So, are there general questions about anything here, or is it time for lunch? All right, it's time for lunch. Good.
So before we have lunch, let's take 1 minute. Can we all go into the lobby? I want to take a group picture.
Ooh.
So